Good afternoon, everyone. My name is Alan Handyside, and as past president of PGDIS, I'd like to add my welcome to you all to this first annual meeting in 2 years. Sadly, I'm not able to attend this year because I'm recovering from COVID, but I'm looking forward to catching up with friends and colleagues next year. Today, I'm going to talk about preconception genetic testing using genome-wide single nucleotide polymorphism genotyping and meiomapping or karyomapping for maternally inherited monogenic disease and meiotic segregation errors which result in aneuploid eggs and embryos. So first, to clarify what we mean by preconception genetic testing. So preconception genetic testing differs from pre-implantation genetic testing, or PGT, because the two polar bodies produced in the two meiotic divisions of the egg are biopsied before the male and female genomes unite at syngamy. And you can see that beautifully in this figure here, in which a biopsy pipette has extracted the first polar body and is about to extract the second polar body. And here we have the two pronuclei that have begun to come together for syngamy, but haven't completed syngamy at that stage. Preconception genetic testing was pioneered by my friends and colleagues, the late Yuri Valinsky and Anne Vakuliev, who is the executive president of PGDIS and was for many years used to test for maternally inherited monogenic disease using STR markers and meiotic segregation errors causing aneuploidies in the egg using FISH. And indeed, they went on to show that you could also diagnose the inheritance of structural chromosomal rearrangements, as you can see here, using fluorescence in situ hybridization to chromosome-specific probes. Preconception genetic testing has fallen out of favor in recent years in preference to blastocyst biopsy. But I would argue it's well worth revisiting now that we have genome-wide molecular methods for accurate analysis of polar bodies. So polar body biopsy then, and meiyomap and karyomap analysis. What are the advantages of polar body biopsy and testing? Well, the first is that it's relatively non-invasive. You're not removing any cells from the embryo. And of course it is invasive. You have to make an opening in the zona, you have to remove the cumulus cells and so on. But nevertheless, it has been proven in routine clinical practice, as I've just explained, for many years in the 1990s and early 2000s. And today, obviously, polar body biopsy, followed up by time-lapse analysis and/or extended culture to the blastocyst, eliminates the possibility that you would test a lot of eggs and embryos unnecessarily, because some of those, perhaps up to 50% of the embryos that you biopsy, will arrest later in development. Now, testing of both polar bodies allows accurate identification of age-related segregation errors in maternal meiosis and indeed single gene defects or monogenic diseases which are maternally inherited. But there's also the possibility of testing the first polar body alone, as we'll see, because you can identify the risk of errors in the first meiotic division, and this could potentially be used for application in egg donors and for social egg freezing to rank embryos for transfer. Once the polar bodies have been biopsied and the whole genome has been amplified, you then have the choice. Well, first of all, you need to genotype the polar bodies genome-wide on a microarray, generally speaking. And then you can apply either meiomapp analysis or karyomap analysis. Meiomapp analysis is actually higher resolution because you're only attempting to detect the two maternal haplotypes, and you typically use a second polar body as a phasing reference if that's available. But you don't have to do that. There are other possible options. For karyomapping, because you're using the same genome-wide SNPs to identify the 4 parental haplotypes, both from the father and the mother, then the resolution is less. It's still nevertheless very high, and you generally use a relative or sibling embryo as a phasing reference to do the mapping. So genome-wide SNP genotyping of maternal DNA and both polar bodies allows detailed examination of chromatid segregation and recombination in both meiotic divisions, and a typical pattern for normal segregation of bivalent chromosomes and then chromatids is displayed here. What you can see is that in the pericentric region, a single maternal haplotype is identified as either orange or green in this display in the first polar body, and the opposite maternal haplotype is identified in the chromatids, which have now segregated and are present in the second polar body and the oocyte. Further along the chromosome arms, there are recombination events or crossovers, and the bivalent chromosome has regions with SNP markers for both maternal haplotypes as expected in the first polar body. Whereas for the second polar body and the oocyte, which inherit single recombinant chromatids with corresponding crossovers, there are no regions identified as having both maternal haplotypes. So this would be the normal pattern in the first and second polar body and the resulting activated or fertilized oocyte for the maternal chromosome. Now I just showed you the normal segregation pattern. Here are some examples of abnormal segregation patterns, focusing on how these can be detected in the pericentric region of each chromosome. In the first example on the left, instead of a single maternal haplotype, either orange or green, both haplotypes are identified, hence the shading in blue. This type of abnormal segregation event we called reverse segregation, and in this example it is followed by another error in the second division, resulting in the oocyte inheriting 2 chromatids, each from one of the bivalents, which would result in trisomy. Now there's not time to go into detail on the other types of errors, but I think it is clear that these can be identified by analyzing the patterns in the first and second polar body by meiomapping. I'm grateful to Pavy Forsblom at the SYN lab in Munich for permission to show this example of high-resolution linkage-based testing for this maternally inherited autosomal dominant condition, spinocerebellar ataxia type 3, or SCA3, caused by mutations in the ataxin ATXN3 gene on chromosome 14. I'd like to highlight two important features, and apologies that in this display the presence of both maternal haplotypes is highlighted in red and not blue. The first feature I'd like to point out is that this high-resolution meiomapp analysis of the SNP markers enables accurate testing even when the disease locus is very close to a recombination event, which could lead to errors or no result if only testing with small numbers of flanking STR markers, for example. Secondly, with oocyte 1, an abnormal segregation event has occurred for chromosome 14, which has resulted in the absence of that chromosome in the second pole body. Again, other methods would not have detected this event necessarily and may have resulted in an error or no call for that particular oocyte. Moving now to SNP genotyping and karyomapping, this is most commonly used with biopsies from embryos, but because it can be applied to single cells, unlike, for example, NGS-based methods, can also be used for polar body analysis very efficiently and accurately. Using karyomap software, which I developed several years ago, the haplotypes of both parents are easily visualized and highlight various features, including meiotic trisomy, monosomies, and deletions. In this embryo sample, there are no less than 4 trisomies identified because they have multiple regions in which the SNP markers for both haplotypes from that parent are present. There is also a maternal monosomy for chromosome 21 identified because the SNP markers for both haplotypes are missing. Karyomapping, although lower resolution than meiomapping, can also be used for polar body analysis. In this example, both polar bodies are analyzed as well as the corresponding embryo. Regions with both maternal haplotypes are present in all chromosomes, which is the normal pattern following the first meiotic division, in the first polar body. And for chromosome 4 in this panel, the pattern of segregation is normal for both meiotic divisions. However, for chromosome 21, only one haplotype is present in the first polar body, probably due to an abnormal segregation event in the first division. Among the other highlighted chromosomes in the panel, there are 4 meiosis II non-disjunction events and 2 reverse segregation events, one balanced by the segregation of chromosomes in the second division and one unbalanced in which both chromatids end up in the fertilized egg. This particular example is an embryo that was an abnormal arrested early blastocyst. However, being able to identify the patterns of segregation errors and their frequency may be useful clinically, so that, for example, multiple errors in the second division may indicate that normal insemination may be preferable to intracytoplasmic sperm injection. So in summary, genome-wide SNP genotyping of polar bodies and either high-resolution meiomapping or karyomapping allows accurate linkage-based testing for maternally inherited monogenic disease and simultaneously identifies maternal meiotic segregation errors of all types. Meiomapping of the first polar body alone allows the risk of segregation errors because it picks up the abnormal segregation events in the first meiotic division. And this may be applicable for egg donors, for example, and in social egg freezing, where women may want to know what their risk of aneuploidy is with the eggs that have been collected, and can even perhaps be used for ranking the oocytes for fertilization and transfer. Meiomapping or karyomapping of polar bodies for aneuploidies of maternal meiotic origin is actually also an alternative to copy analysis of embryo samples, and of course completely avoids the detection of intermediate or mosaic copy number changes of uncertain clinical significance, which result from analyzing multiple cell blastocyst biopsies. Thank you for listening, and if you have any questions, please direct them to this via my website, preimplantationgenetics.com, using the inquiries email, inquiries@preimplantationgenetics.com. Thank you very much.